Okay, thank you for inviting me here. I hope I will be able to give some insight on this DNA stuff that is perhaps not very familiar to most of you, but I will not go into details, I want to say that. So if you want details, you can ask me during the questions or maybe later about the details of how this work has been done in the lab, I mean. So this is something that started in 2015. It's a collaborative project between me that works at Uppsala University with people from Stockholm University. And we wanted to use some center of excellence that we have in Stockholm that is doing this new generation sequencing. So they wanted to collaborative work between the scientific world, especially in the geological part of the university, and the Center of Excellence. And they want them to use these platforms that they had. So what we wanted to test, and this was actually the title of the project, to test the utility of the shotgun sequencing technique on ancient lake sediments. And we wanted to look if this is a technique that allow us to have a better view over the entire communities that is living in a lake through the time. And so we wanted to look at both aquatic and terrestrial flora, but we wanted also to look at the fauna and the microbial community in the lake. And this is the site that we have been investigating in southern Sweden, Hasseldala Port. It's an ancient lake. Today it's a peat bog. And here we have a close-up, but we don't see very much, but this is the lake. No, this is the Hasseldala Port, and it is a bog, but it was an ancient lake, so it was a lake before this. The light brown regions here is old peat bog, while the light blue is lakes around. So what we have done, why I'm saying that this was a lake, this is what it looks today, and this was a lake probably in the late glacial period, and that has been infilled through the sediments moving from the lake catchment, and so we have an increased filling of the lake and the basin, and then submerged plants start growing along the edges of the lake, and we have accumulating, and in the end we have I cannot really move without this here. No, you don't hear me. No. I guess. No, okay. So and then we have so peat bog in the center here, and perhaps I can see it better here. Okay, and then grasses and shrubs growing around the lake here and here. So what we have done is we have core, using a Russian core, right in the middle of this peat bog. And, okay, thank you. But actually, if I can read here, it's okay. It's okay, thank you. Because I don't see very well from here. So we have collected a core right in the middle of the peat bog with a Russian core equipment, and we extracted a sediment, a core of this size, and it's long around 1 meter, 1.2 meters, I think. And because Barbara was just interested in this specific period of time, that it's the late glacial, from the late glacial to the beginning of the Holocene, so we were able just to core just this part of the sediment. And this is what we see here. So what we have done, we have looked at DNA. And why do we think that there is DNA in the sediments? Well, of course, there is DNA on the sediments. In the sediments, there is a lot of DNA coming from animals, from plants. So we have cell from animals, cell from plants that are falling in, sedimenting in the sediments. And we can have both extracellular DNA or DNA that is englobed in the cells themselves. And the extracellular DNA, you may think that it's unprotected, but in reality it is protected because it usually binds to the organic and mineral particle of the sediments. And this is a way of protecting DNA, and this is a way why we actually find DNA, even ancient DNA in the sediments. We can extract DNA from sediments from permafrost and ice, even ice cores actually. We can extract DNA from peat and normal sediments, or we can extract DNA from lake sediments or from marine sediments. This has been done in many, many different studies now in the last perhaps 10 or even 15 years. There is another way of protection of DNA that has been proposed, and this is the fact that we think that apart from being released, being protected in the cells, being extracellular or protected by the mineral organic soils particles, it can be also transformed, so inglobated in the cells of the microbial communities. This has been found in some type of sediments. And of course, the DNA that is inglobated in the microbial cells can also be protected through time in this way. Of course, we have to extract DNA from both the sediments and from the cells, either plant or animal cells or bacterial cells. So what we know today is that the mineral organic component protect the DNA, that this reflect local plant and animal community. So if we find DNA at a certain spot in the sediments, we know that this is local. And this is very important for the paleoecological type of study that we are doing, because we know that we are always looking for local signals. And another thing is that if we can extract this DNA and then we can sequence, we can demonstrate the local presence. Of course, we can never say anything about the absence because we have a lot of bias in this type of analysis. So whenever we don't find anything with DNA, doesn't mean that it's not there. There might be many reasons why this is not there, but we can for sure say that it was present. And another thing that we have to keep in mind is that DNA, no matter what type of sediments, what age, it's always damaged and it's always fragmented in short pieces. So what are the techniques that we can use to extract DNA from the sediments? There are two main techniques that nowadays are being used, and some of the people will show the results based on both techniques. The first is that you amplify, so you reproduce the DNA, particular segments of DNA that you are specifically interested in, and this is what we call here barcoding. So you amplify and then sequence. But you can do something else nowadays, and this is called shotgun sequencing. Shotgun sequencing, it means that you do not amplify first the target regions that you are interested, but rather you sequence the whole remains that are present in the sediments. So all the short reads that are present in the sediments can be sequenced directly. And this can be done with very powerful platforms for sequencing that nowadays are available. So the shotgun sequencing approach has a lot of pros, and one of the main pros, probably advantages, is the fact that you can run really, since you get a lot of data, really billions and billions of data sometimes, or millions, depending on what you are looking at, you can look at them from a statistical point of view, and you can, for instance, look at some type of damage. I say that all the DNA in these sediments is damaged, usually, but there is a specific type of damage that we usually find in ancient DNA, and this is a C to T substitution near the fragments, the short ends of the fragments, and this can tell us that the DNA is actually authentic because it's damaged. So if you have contamination from modern DNA, you actually can recognize rather easily. And of course, there are some disadvantages as well, because we have the problem that we have a lot of bacterial DNA normally compared to the plants and animals. If we want to look at animals and plants, then we have a biased data set. And we have problem to assign these very short reads, because I said it's very fragmented. We sequence everything, but these short reads must be assigned. So we need a bioinformatic part that is very complicated for assigning this. So, as I said, this was the study aim and the aims of our project. So what we usually look at in this type of studies is we try to combine different proxies. So if we look at the flora, we try to combine the pollen, the macrofossils that paleoecologists normally get from this type of sediments. We combine this information with the DNA information. Now I put this Venn diagram here showing how the proxy may combine, but this is really, it's just an idea. It changes a lot in terms of size of the circles and how do they overlap depending on the type of sediments we are looking at and the technique that we are using. So you should not take this as an example. And then we wanted to look at fauna as well. So in these sediments, we looked, this is something that I cannot show you results. I will show you just some preliminary results we have for the flora and some also results that we have of the microbiota as well from this lake. Nothing from the fauna because this is ongoing analysis. So we have some ecological question here. Barbara, that is the paleoecology, Barbara Wolfert, the paleoecologist involved in this project. She was interested in one particular time period, and this was the late glacial. So she was interested in see how immigration of different plants and animals and succession of organisms occurred during this specific period. The period is between 15 and 11,000 years ago, and is a period of really dramatic environmental change. And this period here, how you see temperature are changing. We have this older dryas that most of people here can recognize, of course, the boiling Ellerod period, so it's a warm period, and then again a younger dryas and increase of temperature very sharp beginning of the Holocene. So we wanted to look at this period. So what we did, as I said, we extracted the core, we opened it in a sterilized condition. Actually, this part first was not done in a very sterilized condition because this is the normal part that actually people are paleoecological scientists are doing. So they are just cleaning before their own studies. Then the core was subsample for these different points for DNA. So I just here in blue is the cold periods. Here, this is the older dryas, the younger dryas, and the warm period here. So I selected these points on the core and subsample for DNA analysis. And the core was dated, but indirectly, because we had lots of previous cores from the same regions. So what we did, and we had as radiocarbon dating, data for the core number 2 and 5. But what we did for this core, that was core number 7, this is a site that has been investigated really thoroughly during the last 10 years by Barbara. So we had a lot of data. So what we do is a statistical correlation of the Loy percentage curves to the master core that was number 4. So we could really date precisely our samples. So this is some pictures from the DNA lab when we were extracting, actually not extracting DNA, but subsampling for DNA extraction. And then here in the ancient DNA lab. So this is the way we work when we work in ancient DNA lab. Of course, we have to wear these funny suits and work in very sterilized condition. It's really hard work. And as I said, we can run some statistical analysis on these very, very short reads that we get from the sediments, and we can, in this way, assure that we are dealing with ancient reads. Okay. So what we can look at, this is so on the axis here, you see that you have an increase here of percentage of substitution rates from C to T and A to G at the end here and at the beginning here of all the reads that we have analyzed. So this one is the increase of substitution of one type, and this is the increase of substitution of the other type at the beginning of the reeds and at the end of the reeds. So in this way, we are really sure that we are dealing with reeds that are authentic and are not coming from contaminants. At the same time, we can look at also fragmentation rate of this. And okay, I will just, since I have very short time, I think I will move on here. This is just the fragmentation rates that we see that we have very short fragments in our samples. And so this is the statistical analysis that we can run on our data, and it tells us that this is authentic. And so what do we get in the end? Well, what we get in the end is really amazing. So we can say that of the whole data set that it's really, really huge and large, we get a large proportion that is actually no hits or not assigned. And this is not because, this is a database, so to say, problem. It's just that we cannot simply, we don't have many of the reads that are in our data in the database. So we have a proportion that is bacterial, archaea, and eukaryotes that has been assigned, and this is just maybe something like less than 10%, or no, sorry, about 15%. And the eukaryotes are actually very few reads, so which means animal and plants, and the rest is archaea and bacteria. So this is archaea and this is bacteria. And if we look at even these tiny parts, that is eukaryotes, the plants part is very little again. So you see that it's a tiny proportion of the whole data set that it actually belongs to plants. And here we have animals, different type of animals and different type of eukaryotes. This is all over the data sets. And this is what we get for plants. And here we can address our ecological question. Do we actually see a change in number of species through the time correlated with the climate change, the transition from the cold to the warm and again to the cold and then warm again? And we actually see this. This is a preliminary data. There is a lot of data here that has been not treated yet, so I have to look at them in detail, but this is just to show you the type of data we can get. And this is not giving any idea of abundance of reeds. This is just the proportion of species that we find. But we see that in the warmer period here, we get a larger number of species detected. And we have, of course, for the same course, a lot of information coming from the paleoecological studies. So we have pollen, we have macrofossil here, and some do overlap with the plant species that we find with DNA, of course. And then as a last one, I wanted to show you the very interesting results that one of our postdocs is working at, and she's looking at shifting communities of archaea. If you remember, I was showing you that most of the assigned sequence are coming from microorganisms, and archaea is a group of microorganisms present in these sediments that it's related with methanogenesis, so they are producing methane. And what she was looking at, this postdoc, she was looking, Angie was looking at the relation in shift of communities, of archaeal communities through time. And here, so we have the course, and here is the shifting communities. So here you have all the most abundant archaea species, and here are at the phylum level. And what she was trying to get is just that she is comparing the proportion of two different main groups of archaea groups, so two mains, that this proportion she wanted to test whether it's related to increase and decrease temperature during this time. And she can relate this, of course, to many other type of proxies that she has available for this core. For instance, this one here, based on chironomids, about temperature, and this is the one that she has that has been proposed based on presence of plants. So we have a lot of data that can be combined with this, and she's proposing this index for archaea communities. for explaining temperature. Probably it's more related to the temperature in the lake rather than temperature, air temperature. And so I wanted to say thank you to a lot of people here that is involved. It's not only Mikkel, Angie, Han, Barbara, Tanya, and Per, but it's a lot of other people around here and behind. But these are the main people involved and that has been doing together with me large part of the work. Thank you.
So it's my pleasure here to present this work, which is a collaborative work between Sarah Schmidt and me from the University of Lausanne, Christoph Sperisen from the WSL Federal Institute in Switzerland, and Willi Tinner from the University of Bern. This method actually allows to identify genes or genetic polymorphisms in subfossils, but it also allows to do the same job in ancient environmental DNA, theoretically. So we all know that we can get lots of information from lake sediments, and lake sediments obviously are a key to understand the past. So this is a picture from the Origlio Lake in Switzerland, where we've been coring our samples for this study with the platform from the guys in Bern, well attached here on the right frame. And so the first type of data we can get from those cores are the pollen records, and as exemplified here, so there is a laser as well, right? It is, yeah. Fantastic, thank you. And as exemplified here in this pollen diagram, we can track the abundance of one or several species through time. Here, this is the record for Abies alba, the silverfear, during the first part of the Holocene. And so you can see that the species has been quite abundant in the community, in the tree community at that time, well, in the plant community at that time, ranging from nearly 40% to a bit less than 10%. And what is quite striking on this chart is that around 6.2 kilo years ago, there was an abrupt decline. So at 6.2, 6.3 thousand years ago, there has been this very strong decline followed by an expansion. And the paleoecologists know that this decline is related to an anthropogenic perturbation related to the establishment of the first agrarian human societies in the Southern Alps. So dramatic change in ecosystems due to firing, burning the forest, to grazing in the understory. And around Origlio, several populations of Abies alba actually got extinct during this event. But in Origlio, well, despite reaching very low levels, very low density, Abies alba could recover and reach again quite large densities before eventually getting extinct during the second half of the Holocene, presumably also because of human activities. So this is something we know from the cores. The other thing we know from the cores is what Laura has explained just before. We can extract ancient environmental DNA. This is a picture from Lars Holm depicting all the different organisms we can get from these ancient sediments using the techniques that Laura depicted just before. And there is a third type of material that is a bit too much underused, according to us, or at least that would benefit to being used much more, which is the subfossil remains, or the macrofossils. So here, you can see those subfossils from, which were taken from this core. So this is a 7,000 years old subfossil needles, or subfossil needle. It's cut into several pieces, but we also get entire needles. And because we were interested in knowing what happened during this strong decline here, we sampled macrofossils before, during, and after the decline. So the abundance is from 0 to about 15. And so, well, the number of macrofossils we got in this single core was realistically good to perform some preliminary genetic analysis, not good enough to make proper population genetics inferences, but still it gives a good information about what is feasible or what is not feasible. So Laura already mentioned that ancient DNA is usually highly fragmented. So on this chart you can see the fragment length here of DNA, and for ancient DNA or historical DNA, the fragments are usually smaller than 150 base pairs, which makes very difficult to apply classical genetic techniques such as amplicon sequencing or rad-seq sequencing, which is quite frequent method used in the context of next generation sequencing. And these two methods need at least fragments above 150 base pairs. Some can be used still with 100 base pairs, but still it's suboptimal. So the same way we've seen with Laura that direct shotgun sequencing can be useful for environmental DNA, we thought, okay, maybe we can also shotgun sequence those needles. And that's what we've done here with several of those guys. Here, this 7,000 years old needle. And first, we were quite happy because on this chart, on the top, you can see in green, this is the percentage of eukaryotes reads from a needle of silverfir. So more than 90%, very few bacteria, a few unknown sequences and viruses. But then, when looking what's inside those eukaryotes, actually more than 2/3 were from metazoan origin, which was quite unexpected because we were extracting DNA from a needle of a tree. And the percentage of reads from land plants or viridiplantae was actually lower than 15%. And more striking, when looking within those plants, only a very small fraction were actually from the family of interest. So this means that actually the needle itself is just a subsample of environmental DNA because it is porous, and all along all those 1,000 years, it gets polluted by all the environment around. So what we get here is actually a good idea of what was around in the community. So in order to go further, we had the chance to know, well, to read some of the literature of our colleagues working on ancient DNA in humans. And there is this Carpenter and colleagues paper in 2013, which was really enlightening to us, and which is entitled Pulling Out the 1% Whole Genome Capture for the Targeted Enrichment of Ancient DNA Sequencing Libraries. So they were facing the same problem as we were facing. They also had 99% of contaminants, even if they were working with bones or with bone samples. The problem is that the method they propose here, whole genome capture, is quite expensive. I mean, usually they find a few skeletons or a few bones, and they analyze the whole genome of those specimens. But as ecologists or population geneticists, we are much more happy if we can work with more, well, with larger population sizes, time series, spatial series. And this is why we have developed a new method, which was published on Monday. It's called HiRAD-X, which means hybridization capture using rad probes, and the X comes from the exome template we use for this technique, which is derived from a first version of HiRAD, which was just called HiRAD. And so I will just explain you briefly how it works. So this is the needle we extracted. We perform shotgun sequencing library preparation the same way that we would do for any kind of sample we use for next generation sequencing. So we have, of course, within this library, the DNA of Abies alba and all the contaminants. So I should have shown maybe only one green molecule and 99 red molecules, but this is just an illustration. On both sides, we bind adapters, but this is just a technical point. And in parallel, we just go into the field and we sample some buds of a fresh Abies alba guy. We extract RNA, total RNA, from which we only take the messenger RNA fraction, which is the exome. And in other words, we're only using the fraction of the genome that is expressed in this specific individual. So it's a very strong reduction of the genome from several orders of magnitude. We do a kind of further reduction using the so-called RAD-seq method. I won't go into details now, but please feel free to ask questions. And eventually, after doing some size selection to even select for a smaller number of reads, of a smaller fraction of the genome, we label bioteins to several nucleotides. So these bioteins are molecules that have a strong magnetic affinity to other molecules called streptavidin beads, or streptavidin in general. And eventually, we combine our library of interest with the probes through a step called hybridization capture. And so the unwanted fragments are filtered out, and after putting the streptavidin beads, we can extract the homologous fragments from our sample interest to the probes, and eventually we can enrich and sequence the libraries. So let's come back to our biological example. So you remember this, with my finger in front of the laser, it works not very well. So this was a decline, and we had samples before, during, and after. And we wanted to test a very simple hypothesis, which is who were the guys recolonizing the area after this very strong decline? Were those guys, those Abies alba individuals, from the original population, and then they recolonized the area through internal recruitment, or were they coming from outside through external recruitment? Were they migrants from other populations? Well, it's quite easy to test this from a population genetics point of view, because if there is no genetic differentiation between the time slice before and the time slice after, one can conclude it's internal recruitment, whereas if there is genetic differentiation, one can suggest that there were some migrants coming to recolonize the area. So, first, we obtained several thousands of SNPs, but we chose the 600s that were the cleanest, and cleanest in terms of population genetics properties, and also the ones that were present in as many samples as possible. And what we found with our sampling size here, as you can see, we have 22 samples for quite ancient time slice, 5 for the time slice just before, 6 for the time slice during the decline, and 14 for the time slice after. What we can see is that allelic richness is more or less stable through time, whereas observed heterozygosity shows a drop during the decline, followed by an increase to the original values or nearly. But when looking at population differentiation, what we find is that there is population differentiation between the time slice of the decline and the time slice after. There is marginally non-significant differentiation between the time slice before and the time slice of the decline, but there is no differentiation between the time slice after and the time slice before, meaning that with an FST, which is the value we use, the estimator we use for computing genetic differentiation, there is no genetic differentiation, and we can therefore conclude that the population expansion is likely driven by local, previously fragmented patches of individuals. So, of course, we don't know exactly where they come from. They could be 1 kilometer, within a radius of 1 kilometer, maybe of 10 kilometers, maybe of 20 kilometers. This we should know soon by knowing the isolation by distance pattern in this area. But what we know is that they did not come from far away.
So you just have 1 minute, less even.
I'm done already? Or?
More or less. Yeah.
Okay, so I will just talk about the perspective we have, which is about comparing these anthropogenic perturbation effects on tree genetics with non-anthropogenic perturbations. Here we have picked up the 8.2 kiloyears cold climate episode, which is due to massive influx of cold water in the northern hemisphere, which has changed the climate due to the disruption of a big glacial lake that was beforehand not really linked with the oceans. So we're now in the process of analyzing those 3 species at the tree line or near the tree line, because during this episode the tree line shifted quite dramatically. So there has been local extinction. And we will check in those populations whether some genes related to adaptation to climate, which has been something shown to occur in distantly related conifers, so adaptation to climate mediated by the same genes in sample in species distant by more than 100 million years ago, if this is something we still see during a very short episode. So thanks for your attention. I'm done. I would like to thank all our institutions, University of Lausanne, WSL, University of Bern, as well as our main funding agency, the Swiss National Science Foundation, and you for your attention.
The libraries that you use, I just wonder, do they cover all plants on Earth? Or because Laura showed this picture that during warm intervals there was a higher diversity than during cold intervals. But is it possible that the plants that were present during the cold intervals are not in the library? I just wonder, the library, how complete is it?
Yeah. So the library is just a subset of all the molecules we have in the sample, but then we enrich this library for a given species, or let's say lineage, by using this hybridization capture. You probably recall this is an important step.
I think I mean the library of present plants and their DNA. How big is that? Because you compare, I suppose, you compare what you find with a library of present plants and their DNA.
So not really. Here,
okay.
Here we just extract genes from the past. So we know from the observation of the subfossil needles that it is Abiesalda. So
yeah, but for other plants.
So if you want to work on other species such as Salix, for instance, I mean this is really feasible. Let's talk about that later. Yeah, sure. Okay, thank you.
Okay. So today in my talk I'm going to present a record we've been working from the Polar Ural Mountains. It's a plant aDNA record from Bolsoi Sucia. And yeah, it's located right in the northernmost part of the Ural Mountains. And this is part of an ongoing collaboration with the University of Bergen, with John Inge Svensson and his colleagues in the Department of Geosciences, who have been working in this area for a long time. So I thought I'd just start my talk by giving just a brief introduction of the work they've been doing to refine the glacial history of the region, and just try and explain why the region is interesting to us in terms of understanding floral changes during the last Quaternary interval. So the DNA work is part of 3 international projects, and I just want to begin the talk just to thank my collaborators in Bergen, Tromsø and Grenoble for this, where the DNA work really wouldn't have been possible without the help of these projects. And the DNA record from Bolsoi Sucia is really only one of a number of proxies that are currently being done on this record. There's Anna Buna, for example, who's doing the pollen, currently working on the pollen record. And there's also been biomarkers, chronomids, and geochemical analyses. And basically our hope is to synthesize all of this paleoecological and geochemical analysis to really build up a comprehensive picture of environmental changes in this area over the late Quaternary interval. So for the DNA work, we really set out to test whether we could successfully extract plant aDNA from inorganic sediments of a large deep lake. Obviously, we've seen the potential of the plant aDNA record on lakes from, for example, the Alps, Svalbard, Siberia, for example. But really, we wanted to push it a bit further here and test whether we could extract plant aDNA from a deep lake with a large hydrologic catchment. And progressing on from this, we really want to see whether the plant aDNA would help us to resolve the floristic composition of the steppe tundra biome in the polar Urals during the last glacial maximum, and later how the floras change in response to the environmental changes from the late glacial to Holocene transition. And overall, we have the aim of trying to understand how the flora has changed in response to environmental changes over the late Quaternary. And this is the hope of giving us a better understanding to try and anticipate how the Arctic flora may respond to future changes in climate. So thanks to Jan Mangerud and John Inge Svensson and others who've been working in the region, we now know that the Ural mountain chain, within the Ural mountain chain and the area around it, hasn't been covered by ice for the last 50 to 60,000 years. Dating of moraines within the polar Urals themselves, the northernmost part of the mountain chain, which I've coloured in a yellow box here, has revealed that this area was not covered by ice since at least marine isotope stage 4. And during the last glacial maximum, 25 to 20,000 years ago, the polar Urals, obviously the Barents Kara ice sheet, reconstructed in the white here, didn't actually reach the northern rim of the mainland during the LGM. And within the Urals themselves, there's actually only a few small cirque glaciers which existed in the polar Urals, and they weren't actually much larger than they are in present day. So the fact that the polar Urals remained ice-free during the LGM kind of sparks interest in understanding this region, in the environmental history of this region, and testing whether certain plant and animal species were able to survive and persist here during the adverse climate conditions of the LGM, and later how they responded to later on the environmental changes as we progress into the Holocene transition. And there have been a kind of handful of paleo records from the polar Urals themselves. I've just marked them in the map with the red markers here. The main thing to note here is that a lot of they're pollen records, a lot of the records are only short exposures. They really only cover a snapshot in time. There are a lot of permafrost sections, for example, or their lake records only begin in the late glacial and cover a part of the Holocene. We're really lacking a record that covers from the last glacial maximum up until the present day. And it's a hope that Bolsu Suchia, marked in the star symbol here, would help us to fill this knowledge gap. And after all, understanding the floristic changes that have occurred in the polar Urals is really important. The region itself is actually kind of a break point between the floras of Europe compared to eastern Siberia. So to understand what has shaped the distribution of those floras in the past, to understand their future distribution, is a really important task to undertake. So this is the Lake Bolsoi Sucha in the polar Urals. It's quite a striking lake in the landscape. It's 12.8 kilometers long and 4 kilometers wide. In its deepest point, it's 136 meters, which gives a perfect opportunity to test whether we can successfully extract plant aDNA from this lake. And a series of cores have been taken over the years from the lake. The plant DNA record I'm presenting here is retrieved from the southern end of the lake, and it's got a very fast accumulation rate. We've got 24 meters of lake sediment, and that covers the last 25,000 years. So for the DNA, we took 153 sediment samples and analysed them based on using the GH primers for the P6 loop. And for every sample, we did 8 PCR repeats, just to increase the confidence in our results and trying to increase the chance of detecting taxa with low quantities of DNA. So the results were really promising from this record. We really weren't expecting such a diverse range of taxa. So after filtering, we retrieved 167 plant taxa, and the taxonomic resolution for this record is really high. We got 45% of the 167 taxa are identified to genus level, 41% to species level, and 14% to family level. And there's some really nice clear switches in plant functional groups across the 24-metre record. I just plotted plant functional groups again as a percentage of total DNA reads here on the figure. And you can see in blue here, this is forbs, and they really dominate the record. Obviously, the main thing to note here is we know from the Yokos paper in 2012 that forbs are overrepresented in the DNA. So to take this figure kind of lightly, but really I put this in to illustrate the really range of ecological habitats we've picked up in the DNA across the record. So in the full glacial zone, around here, we get obviously a lot of Arctic alpine forbs. We get a lot of grasses as well, and there are a few occurrences of trees. That's mainly elm actually, and there's a lot of work still to be done to figure out why that's occurring. But then as we get into the late glacial, the bryophytes in the red do increase, along with first gets in the taller shrubs, the alder, for example, and the shrub birch. And as we go into the Holocene, we then see the appearance of trees and the small and the dwarf shrubs, for example. So because the record is so diverse, the only downfall is we don't actually have time to go through the entire species list. So if you do want to see the list, please do come and grab me, and I can show you the full species list from this record. I've just put here kind of a summary diagram of some of the key taxa we found over the record, and you can see the clear switches in different functional groups. So the plot here is presented as the number of PCR repeats with more than 10 reads, and it's against depth here. And you can see the radiocarbon ages plotted on the left-hand side here. And in the DNA, we've identified 4 zones. This first zone is the full glacial zone, and you can really see the dominance of grasses, particularly some salt-tolerant grasses such as Puccinelia here. We've also got Festuca. We've got, main thing to notice, we've got salix throughout the record. It's common in a number of other lake records. But we've also got a lot of Arctic alpine forbs. So we've got Papava, occurs throughout the record. It does decline in the topmost Holocene zone. We've got Draba, Anthemidae, Paria, Oxyria, and stuff like that. So yeah, there's a lot of diversity, even in the full glacial, which you really don't see in pollen records. We really don't see as much kind of the forb diversity in this. So the DNA is really kind of adding to the picture of this region. The second zone actually correlates with the bowling arrow rod, and I drew this zone basically on the signal from Carex. It really starts to rise around here, the late glacial interval. And we also get some other forbs that really appear in the late glacial and carry on through the Holocene in this zone. And the third zone here is actually, I suppose, a younger Dryas interval. You can see the Betula curve here really dips in this zone. And there's a few like Paria, for example, has a rise, and there's a few Breum appears in this zone, but really birch is really the only taxa to show a really clear signal to younger Dryas cooling here. And the fourth zone is the Holocene. This is really where we get the appearance of tall shrubs, dwarf shrubs, and the trees. So the spruce here, you can see here, does appear slightly earlier than the larix in this record. And at this time, we also get alder coming in and some woody shrubs such as Petra and Vaccinium, got Arctostaphylos is in that record. And we really get, there is turnover within plant functional groups. You can see in the grasses here, we've got a real turnover from these full glacial grasses to then the appearance of grasses such as Agrostidinae in the Holocene, and kind of the decline of other taxa here. So it is a really neat record. Because I tried to summarize these the vegetation that's happening because we've got so many taxa. So I've drawn a series of cartoons to explain what is going on in the record. So the full glacial zone is really dominated by the Arctic alpine forbs. So we've got a lot of kind of classic tundra, some steppe species in this zone. We've got a lot of papaver, draba, anthemidae, it's probably Artemisia, and a lot of saxifraga species. We also do have salix shrubs, like throughout the record, they are present, and a lot of grasses that you don't see the diversity of grasses in pollen so much. Maybe some mammoths as well. We have actually currently running the record using the mammal primer, and the results should be back in the next few weeks. So we'll have to wait and see if whether mammoths were there or not. We shall see. So anyway, the late glacial zone, we really start to see the appearance of more moister taxa. So this is really where the carex kicks up here. And we also, some other forbs start to appear, like Myosotis, Erytricium, Bistorta, Caltha, for example. And in the shrubs in the late glacial, now this zone, just to note, this zone includes the bowling alarod and the younger dryas. So Betula appears here, but we also get that decline in Betula in the younger dryas. But we also get a lot of the grasses, again, are still present in this zone.
So 1 minute.
Thanks. And then here I've split the Holocene into 2 zones, and this mainly because it's the Holocene thermal maximum, where we get a lot of trees. We've got spruce and larix and alder shrub. Just to note, particularly Betula, it could be shrub or tree Betula. With the DNA, it's only to genus level, and we really don't know. Here on my cartoon, I've kind of exaggerated and suggesting it's tree birch. But we also get a lot of, again, the woody shrubs. But the main thing to note is the really exciting thing for us is that pollen, obviously the pollen records tend to be dominated by the trees once you get into the Holocene, the high pollen producers. But in the DNA, we've got a lot of Arctic alpine forbs as well, which is really exciting. And the last one is the late Holocene, where we see the decline of the trees, but the reappearance of the kind of re-establishment of the woody shrubs, such as Impetrum, Vaccinium, Arctostaphylos, for example. And this is the appearance of the other grasses, for example, as well. Just very quickly, I wanted to compare how the DNA record looks compared to the regional vegetation history. So this is a comparison to 3 of the pollen records we know from the region. And just to note, we understand they are different source areas, so we wouldn't expect them to be exactly the same. But there are some really neat trends in the DNA data, particularly the arrival of trees. And the full glacial zone. I've coloured the actual DNA record slightly different colours, just to represent that they're the similar zones, but we get a lot higher diversity in the DNA. So obviously, particularly in the Holocene, the pollen records are dominated by the trees, but we've also got a lot of forbs as well in the DNA. So just to conclude, for the DNA, we've got very high floristic diversity over the past 25,000 years for this record. It's got excellent DNA preservation, and we've got kind of a DNA signal from a range of ecological habitats, which is excellently preserved in such a deep, large lake. And this is probably likely due to we've got very steep slopes of the catchment. There's got a real input of inorganic material. We know from past studies that the DNA tends to bind to kind of fine sediment particles, and this core is really dominated by silt and clay, for example. So maybe that is a factor in why we've got such great preservation. And lastly, we've got a really high floristic detail from the DNA, which really we don't have from the current pollen records from the polar Urals. Thank you. Thank you.
My presentation is about sedimentary ancient DNA and how it offers new insights into the vegetation history of Western Beringia since the Eemian. The late Quaternary was characterized by pronounced glacial, interglacial cycles, and those cycles had strong effects on global temperatures, ice cover, and sea level, but also on biodiversity, and this resulted into spatial shifts of major ecosystems such as tundra or taiga. And in my talk, I want to focus on the past 120,000 years from the, how does this work? From the Eemian interglacial to the Holocene interglacial. Climate models project an increase of the mean annual temperature for the Arctic of about 5 degrees Celsius, and this is what this matches the reconstructed mean annual temperatures of the Eemian, that was also about 5 degrees warmer than today. And the problem is that we don't, that Eemian paleobotanical records are highly underrepresented, and one of the reasons is because of the global sea level rise that started in the early Holocene. So vast regions of the former landscapes are now inundated by the sea, and here, for example, I show a picture of, or a map of the Laptev Sea region, and most of the former landscapes are now the shallow shelf systems, and only a few islands reach out of the sea. So these islands are very valuable paleo archives to reconstruct vegetation history. And our aims were to add new paleobotanical records for the Eemian to close that gap, and to using sedimentary ancient DNA as a proxy. And furthermore, we wanted to track changes of the terrestrial vegetation. In particular, we wanted to track the changes of tree and shrub taxa since the Eemian. Our study area is Bolshoi-Lyachovsky Island, and is one of the New Siberian Islands. This is one of those islands which were reaching out of the sea, and today the island is covered by tundra. It is treeless and dominated by grasses and sedges, as it is shown in this picture. We took 4 permafrost sediment cores along the southern shoreline, and those cores that were recovered ranged from the Eemian until the Holocene, only with the last glacial maximum period missing. To subsample the cores, we went into a climate chamber and sampled at -10 degrees Celsius to prevent thawing of the material, and we removed several layers of the outer sediments to drill out a piece from inside the core. And then we isolated total DNA in a special ancient DNA lab, also with the funny suits, and we isolated DNA from 72 samples, and we also set up our PCR reactions with the TRNLP6 loop marker in the ancient DNA lab. Then the next step is the sequencing on the Illumina HiSeq. So it is a DNA metabar coding approach, and afterwards we use the OBI tools for bioinformatic filtering and to assign the taxonomic names. Then we also include a final filtering step, so we only keep those taxa that have 100% match to the sequence, to a sequence database entry, and they must have at least 10 read counts per sample to be considered further. We also removed exotic taxa that are not from the Arctic, such as Musaceae, and we also removed aquatic and swamp taxa because we wanted to get a better signal of the terrestrial vegetation. So in this plot, you can see the number of taxa for each of the 4 cores, and for each core we have increasing depth from top to the bottom, and in dark gray you can see the rarefied richness, and in light gray the observed richness. And in total we detected 320 potentially authentic taxa, with 115 detected on species level and 126 on genus level. We detected the richness was between 1 and 54 taxa per sample. And what we also could see that is that in warmer phases, such as the Eemian interglacial and the early Holocene, we have a higher taxonomic diversity, but the rarefaction had a rather strong impact because we had a high number of rare taxa. The second aim was to investigate the terrestrial vegetation changes, and here again we see a stratigraphic diagram, and in comparison to the last talk, here we don't have the zones, but we have for each core again increasing depth from top to the bottom. And what we see is that for many samples we don't find any tree or shrub taxa besides Salicaea, but for some samples we find a considerable proportion of shrubs and also tree and tree taxa. So here we have a closer look at only the tree and shrubs, and we see that during the Eemian we find Populus, the deciduous Larix, and evergreen Picea. We also find Betula and Alnus, but it's not sure if they are in tree form or in shrub form. And we also find Cornus, and this composition implies maybe an open, but we don't know that, but mixed coniferous broadleaf forest. Then in the early Holocene, we find only large, but we find also Betula and Alnus in considerable proportions, and this is a pattern that we often find in the proximity to the tree line. What we also find is Pinus, but Pinus occurs only in samples that are dominated by taxa that point towards cooler conditions, and there are no other tree and shrub taxa. So the occurrence of Pinus in our record is questionable, but we don't really know if it's from contamination or long-distance transport. And if you have any ideas, please feel free to contact me and discuss that. But Picea and Larix are supported by pollen analysis and stomata. So Bolshoi-Liachowski Island transitioned from mixed forest to treeless tundra, and so what changed? First we had a cooling after the Eemian, so probably it was too cold for Picea and Populus, and those species were outcompeted by Larix. And second, we see that at the early Holocene there was starting the sea level rise, which disconnected the island from the Siberian mainland and led to increased humidity. So the main messages for you to take home is that the vegetation changes were affected mostly by global temperatures, by sea level, and the connectivity or disconnectivity with the mainland. And second, that the Eemian vegetation included evergreen Picea, and this supports climate vegetation models. So if Picea will maybe sometimes be a part of the future vegetation, then we have to consider that it has a strong impact on the albedo, and with this also a positive climate feedback, which should be then also considered in climate models. So with this, I want to thank you for listening, and I want to acknowledge my colleagues who helped me in the lab and by subsampling, or who gave me these nice photos. Yeah.
I was super impressed with your data, basically because, it's a quite old record. Yes. 120 kilo years is a lot. Yes. Did you see any changes in preservation or taphonomical issues in your records?
Yes, what I didn't really show is that several deposits are only or can only be found in under special deposition conditions, such as maybe ancient lake infillings, and sometimes we have ancient active layers. So especially in ancient active layers, we have a very low recovery of DNA, and yeah.
Okay, another question? Yes, Tony and Nadir.
I'm just wondering, what is the chronology? Sorry, what is the chronology based on? Because you could move some of these around a bit, especially sort of 5.
Yes.
So.
Do you mean if we did
method?
AMS or?
Well, it's pre-AMS if it's.
Yes, we did AMS for the younger samples, and the OSL dates are still at work, and also IRSL, but we have many outcrop datings, so we know the approximate chronology.
Right, okay, so that will come, because that will make a difference to some of the interpretation, maybe.
Yeah.
Thank you for organising this session. This talk takes us back to understanding what we're actually finding in DNA results. As a paleoecologist, I'm imbued with a long history, decades of research into finding out what a pollen diagram actually means. And as with pollen, DNA can come from different places under different circumstances and reflect different parts of the landscape. So in the EcoChange project that took place a few years ago, part of our work was to try and find out what DNA in sediments actually means. And here I'm going to focus on the kind of DNA actually just reported now, coming from what is basically a soil sample or a loess deposit or something like that. What we did was work in the Arctic, in Norway and Svalbard, and ask some questions about what's the source area of DNA and how complete is its representation. And the main model that one might construct is that obviously in a soil or a sediment, the DNA is going to be derived presumably from plant material decaying both above and below ground. The sample could be coming from a different place on the landscape, and we might think, for example, that in an Arctic environment with a slope, you would get overground transport through gelifluction or runoff, movement through the active layer. A sampling site here might well have more different kinds of sources of DNA than one here. There's also the issue of stuff coming in, which was just alluded to in the previous thing. Does pollen come in and actually contribute? Do bird droppings or animal droppings coming from a long way away contribute to what's in the soil? So with that kind of model, we're going to try and find out what we actually do see, where it does come from, and then how could we apply this kind of soil-based DNA analysis to some relevant questions. Just to go back, the first study that we did, which has been published a while, asked some questions about whether we could distinguish major patches in the landscape from the DNA retrieved in the soil underneath those patches. This is a landscape in northern Norway that comprises meadows and upland heaths. Those were the two things we particularly looked at. So on this ordination here, the solid circles represent different plots, different 15 by 15 meter plots, where we're in the meadows, and then the dotted circles are a whole bunch of points, samples taken from the soil, analyzed, and then plotted on the same ordination, which shows actually it differentiates quite nicely at this kind of scale between the different patches. So the ecological information in the DNA was telling us about patches on the landscape, which was encouraging. The other thing we did was use the read numbers to see whether there was a relationship between the amount of vegetation on the landscape and what you got out of the reads of the DNA. This landscape's quite big. The plot scale is quite large. The correlations are statistically significant, but fuzzy. And as Charlotte mentioned out, for soil, forbs are overrepresented. Although we know very much less, it's being worked on in terms of lakes, which are harder to calibrate because you've got to measure more vegetation around lakes and you have much bigger catchments potentially. But so we have these relationships that we can apply at least as a sort of rule of thumb when interpreting the data. So the study that I'll focus on here was in Svalbard. It was in two well-vegetated valleys in the sort of inner fjord area where the climate is quite good as far as Svalbard is concerned. And what we did was a very intensive botanical survey based around plots where we took soil samples from the DNA. These were taken with a sterilized trowel, popped into sterile containers and taken off to the lab, and then the inside of those samples was then sampled for DNA and processed accordingly. But the vegetation sampling was done intensively out to 4 meters away from where we took the sample points. And so we have, in this case, cover abundance of everything, and either 3 or 1 sample were taken in the plot. So the procedures worked fairly normally with using the GHP6 loop primers, sequencing using the OBitools set, and also filtering to quite an extent where we didn't in this case have replicates initially. And then subsequently we did the whole data set again with replicates as described by Charlotte to try and sort of get a better verification of our answers. What we did though was have a very rigorous cutting out of things if they didn't occur in one of the samples, if they didn't occur in more than, when we had replicates, half of the replicates at least. Of course, also we did throw out things that represented lunch, and apart from that, actually, with this filtering, we got a flora that was highly consistent with what grows on Svalbard today. So in terms of what was found, almost everything in the vegetation surveys in those plots was what showed up in the DNA, and even more interestingly, almost everything came very close to the sampling point. So here's the vegetation, here's the DNA, 88 species, taxa recorded, and the overlap with the vegetation pretty good, but it's clearly undersampling. So we've got good fidelity, what's there was really there, but the sampling, it lacks the full definition of the species richness. So just to put this in another way, almost all of those 88 taxa were found as a plant growing in the plots, only in the middle plot, so about half a meter to a meter away. It was much less probable that vegetation that was occurring in the outer rings only of those plots would show up in that DNA sample in the middle, as can be seen by this diagram. So concluding from this calibration, if we assume, and there may be some arguments about that, that a soil taken from the surface of the Svalbard landscape is probably similar to something like coming from those Jedema samples we just heard about, or a paleosol, you're basically plopping down a 1.1 meter quadrat and asking what's in it, but you're losing some of your information. You also have a time scale because your sample has depth and thus represents some time. Our macrofossils in this study were typically modern or recent, and we think we've got not a particularly long time span here, and it was certainly enough to give us DNA floras that were basically replicating the tundra mosaic that we measured. And for example, if we were in a plot with vaccinium, a very unusual kind of patch, we found vaccinium DNA. If we weren't in the plot with vaccinium, we didn't find the vaccinium DNA, and that was a pattern that repeated quite a lot. So one other point about presence and absence versus reeds. So this is a similar Jedema section to the one presented in the last talk. This is Devani Yar in northeast Siberia. Ice wedges, and these are the melted out sediment columns, which can be leveled across the landscape, and samples can be taken. So basically, imagine yourself on the top with a quadrat, taking samples up here on the surface. What we're doing down here is the same thing with our DNA samples, our 10 to 20 milliliters of DNA samples being taken out of this permafrost cliff. And what I want to mention is that you can either count the numbers of motus or molecular units, the taxonomic units that come out, which range from species to family, or you can count reads, the number of sequences, and make a sort of abundance diagram. So just taking the two important functional types from sort of, stage 3, the graminoid group and the forb group, going downwards, these are just two samples. And so if we just take the presence of motus, we can see that the graminoids are actually quite predominant. If we now do some taxonomy and nest any molecular unit that actually includes another molecular unit, so if we have Poaceae and then we have Poa, we're going to pop them together, we get a sort of balanced, floristic message of the types of species present. If we do reads, then the forbs start to win. But we must remember that in our analysis, the forbs are overrepresented. So we have to take that into account. So what about some applications? So going back to a data set taken from the EDMA, originally published by Villesleff et al., this is a look using both counting motus and counting reads. So we're doing both a floristic count and a sort of abundance count of different features. And I'm not going to do too much more, except we do see the forbs dominate in the LGM, in that middle sort of stage, late stage 3 period. But as we get to the sort of Holocene period, we see changes with more trees and shrubs being represented in the samples. But really, I just want to focus on not so much exactly what these mean or where they come from, but what you could do with samples like this, because we could talk quite a lot about what these actually mean. So what was done here was looking for some traits that might be interesting to trace how they change from the Pleistocene to the Holocene. And in this particular case, this is Marimora et al. In review. We looked, for example, at mycorrhizal traits and also end-fixing traits. So here we have a huge number of samples. I think this data set has about 250 samples in it from across Siberia and a few other places. So we're really lumping tens or hundreds of samples together, and we're putting them into periods like the last glacial maximum or the post-glacial period. And we're saying what has changed on a bunch of quadrats taken across Siberia, and we can argue about how relevant that actually is. But we see changes, and we see changes, for example, that we might expect, which is the end-fixers in the flora increase during the Holocene, because as carbon availability increases during the Holocene, nitrogen becomes more limiting. Being a nitrogen fixer becomes a good thing. And so we can see these things, and with our potential greater taxonomic resolution than pollen can give us, in the right kind of environment, we might be able to do quite nice trait analysis, not necessarily for nitrogen fixing or mycorrhizae, but something that could tell us useful things because of the taxonomic resolution. Another place where this kind of sampling of a meter quadrat would be exceedingly useful would be for understanding specific questions about archaeological sites. So this is the Upward Sun child burial site in Alaska, studied by Ben Potter. And one could see that if one was carrying out the excavation and you were taking DNA samples at the time that you were running the excavation, you could find out a lot of information about resource use and what was going on that archaeological site. So this is another potential really good application for this soil-based DNA. So to conclude, bear in mind that if you're doing this kind of work, you're basically taking a quadrat and throwing it and finding out what plants are there. Probably the species richness is going to be underestimated compared with what was actually there in any one plot. Therefore, the more samples that are taken, the better, because species area curves work, and the more repeat samples in a small area you can make, the more likely you'll get your full species tally or your full taxon tally. So it's good for local scale reconstruction, and I would say that we can't always assume that all DNA is local, and as Charlotte's study showed, that stuff was coming from all over a huge catchment of many square kilometers. So we've got to think about the environment in which we're taking the sample and what the depositional rules are in those sites. Location-specific reconstructions, coprolites, of course, very interesting, burials and so on. But if we want to say something about the regional picture, which was what the Willis-Leff et al. Paper and the Mora et al. Papers say. We do need to argue that we're in the right place on the landscape to be zonal. So these Jedema uplands are probably representing the zonal vegetation of the time, but if the samples all came from river valleys, then we would have a different story to tell, because our plot would be down in the river valley and it would be telling us about that. So several projects funded this. Several people spent a lot of time on their hands and knees on the tundra in Svalbard, and Heather Binney tirelessly processed data. Eunice helped us, and particularly Marlena and Idun were really good at picking up flags. That's Inga's two girls. They're a bit bigger than that now. Thank you. That's a good question. We actually took the samples by two methods. One was either to take a sterile tube and just press it. We cleared off the top with a sterile bleached trowel, so it wasn't vegetation on the top. And then we just gently pressed a sterile tube in and capped it. And then the material would come from the top of the tube, which would be the top of the soil. In other cases, we actually dug out monoliths and took them back, and then took out material near the surface. It's a good question, and we certainly have time depth in that. Yeah.
Welcome to my talk, and thanks to the organizers for giving me the opportunity to present my research here, and also the research of my co-authors that for some reason, and I have to sincerely apologize for that, are not listed in the abstract book. So of course this work would not have possible without these people and others who helped me along the way. So I have to see if I can make up for that somehow. So I'm an evolutionary biologist, and I'm interested in diversification and speciation, how that interrelates with ecological adaptation in cichlid fishes. And so an approach I have taken in my past work, and that is quite common in the field, is to go to an environment, to an ecosystem, and you see if you find your group of organism of interest there. And for example, in my case, I catch the fish, I take a DNA sample, I sequence that, and then I build a molecular phylogeny. And I use this molecular phylogeny to make estimates of the diversification rate, of the pace of certain adaptation, how they happened in the history of this clade. And that works particularly well if you have a clade that is endemic to a certain environment. Then you can basically, you have a good guess what the maximum age of this clade is. What you will also find is that there are some not so closely related representatives of the group you're studying, for example, not closely related fish in my case. And that is a bit more difficult because you don't really know, you cannot date when they appeared, when they, for example, colonized this lake, and how that interacted with the diversification of your group of interest. Also, if there were recent introductions, for example, human-mediated, that might have outcompeted species in your group of interest, and that happened before records were kept, then you're oblivious to this, and that might actually affect your estimates. Of course, that can also happen in the more distant past. If there were extinction events, it's very difficult to estimate the existence of these extinction events and how they affected the diversification of the clade. So for all these scenarios, it would be fantastic to have, in my case, for the cichlid fishes, fossil data. And in the lakes that I'm studying in East Africa, we have a source for this, luckily, which I'm now studying, and what I will present to you here. The sediment that accumulates over the lifetime of this lake. And of course, to you guys, I don't have to explain that too much, but to my evolutionary biology colleagues, I have to go into a bit more detail there. Yeah, that's what I'm looking at in my cichlid fishes. And these cichlids, let me just introduce those. They are very species rich. They're a famous model system in evolutionary biology because of the species richness that they have accumulated in a very short amount of time. And they've adapted to a large number of ecological niches. They have produced phenotypic adaptations to deal with a whole range of different food sources. And this is not an untypical scenario in an East African lake, for example, here in Lake Victoria, where you have a high density of these fish, and they represent a sizable amount of the biomass in this lake. And they also are, as I said, very species rich. So what does that mean? So we have several hundred species in the larger lakes. And in total, they sum up to about 2,500 species, also counting the ones in South America and Latin America, which is quite a proportion of the total fish diversity we have on Earth, which at the moment is about 30,000 species. So it's almost 10% of that. But you also have lakes that are quite large, like Lake Kivu, which are kind of depauperate in comparison with the other lakes. So yeah, I'm very interested in this system, and I would like to add a more paleo-focused perspective to that. And I get my fossils, I get my fossils from sediment cores. For example, this is just an example from Lake Chala, which I also study with the help of my collaborators. You, of course, know how a sediment core looks like. And I can either pick out the fossils directly if I see them in the section, or if I take a subsample and sieve that, I can find them. And they can be quite numerous, and they're very nicely preserved often. So I can even compare them morphologically to modern samples and learn something from that. But what would be absolutely fantastic now is to generate genetic data from these, because what we most often find are bones that are not diagnostic for certain species, because they simply are, for example, vertebrae, which are seldom bear adaptations to food resources, for example, but also because I'm looking at very closely related species, and they are hard to distinguish even in the more adaptable bones. So here, ancient DNA would really help us to get a better picture. And the main problem that has been raised, or that has been discussed in the talks in the symposium already, is the degradation of DNA. So for example, a whole DNA strand, then the older it gets, it breaks up in smaller pieces. Also, we have base modifications that make it a bit harder to assign a certain DNA fragment to its original source. And unfortunately, this process is not only increasing with time, but is also quite temperature dependent. And that's unfortunate for me, especially, as opposed to most other speakers' work, because, of course, my fish occur in the tropics in East Africa, and this model of the likelihood of preservation of 25 base pair long fragments, so really short fragments, for a time span of only 10,000 years, leaves little hope to actually find ancient DNA in the samples that I'm studying. So yeah, that's a bit unfortunate, but recent studies have shown that it also is very much dependent on the depositional environment and on the actual tissue that you use if you're successful with ancient DNA extraction or not. So I went ahead anyway and had a first trial run at the University of Oslo, and then several more in different labs in Switzerland. And these are actually the bones in there, nicely crushed up, that you've seen before on the picture. And so just a very quick overview of my approach. I used a very standard extraction method for bones. I did a double-stranded library preparation, and then sequenced that, shotgun sequencing, on Illumina platforms. And then I used reference genomes from cichlid species that we have to map my reads to them. I did different analysis using BLAST to look at more specific gene regions, and also Clarke for metagenomics. And yes, I did find cichlid DNA, so that I'm really happy about it, and I was a bit surprised even by the unexpectedly large amount of endogenous reads. I would have expected less. The method works in principle also for these tropical samples, but the success also seems to be correlated with the age of the samples. So for those few samples that I have analyzed so far, I find a strong correlation with the age of the samples. So this is just a relative measure of how many cichlid mitochondrial reads I have compared to other mitochondrial reads. But this might also be confounded by the lake that the fossil is from. So I hope to refine that more with additional samples. Just to outline 3 sub-projects that I so far looked at, or 3 lakes. Lake Kivu is an old and deep rift lake. It has an interesting water chemistry. It's very rich in methane and CO2. But it also has very few fish species, so surprisingly few given its age and size, and compared to the other lakes in the region. One idea that we had was if maybe catastrophic degassing events led to the extinction of a large amount of a possible endemic cichlid fauna that had been there before. And we wanted to test that, or see if we could test it with fossils and ancient DNA. So I took these 2, so this one assemblage of fossils and another sample from a bit more down core. I made a reference alignment, so I pulled diagnostic SNPs from a reference alignment of the ND2, mitochondrial ND2 sequence from the modern species that we find in Lake Kivu. So one big clade, just 16 species, I think, in total. They belong to the Lake Victoria region superflock. And as you can see, they're very closely related. So they basically have no diagnostic SNPs between them in ND2. And the second clade that we find in Lake Kivu is one ancestor of this superflock, the Upper Nile lineage. And there you can see you can actually find diagnostic SNPs in the ND2 sequence. So if I now compare my ancient DNA reads to that and see where I have coverage, which is of course not every site, but enough to actually assign the fossil to the Lake Victoria region superflock. And I know that it's not the other group that's also present. And the same for a slightly older fossil, where I have higher coverage. So we recover the entire sequence of the ND2 diagnostic SNPs here. So we do not find evidence that there has been a different lineage present, but we've shown that the method works. So adding maybe older fossils and higher coverage and more of them, we might be able to thoroughly test that hypothesis. A different project was in Lake Chala. It's also an old lake and deep. It's a crater lake. And currently it's inhabited by 4 fish species. And 3 of those have only recently colonized it, so within the last 70 years or so. And there have been sporadic surveys, so that's how we know these 3 species weren't there before, or at least we have some evidence for that. But it would be nice to corroborate that, and also to see if maybe due to the colonization by these new species, potentially older species have gone extinct. So here are the 4 cichlid species. It's representatives of 3 different clades, oreochromines, tilapioines, and haplochromines. And this is the putative native species. So this is Oreochromis hunteri. If we look at the modern fauna, if we look, for example, at the red dots, this is a haplotype network. So we look at the diversity of mitochondrial ND2 sequences here. We see that we have some diversity, so there are different haplotypes present, with one haplotype being the most abundant. So that indicates that there's some diversity, but also in the other Oreochromis species, for example, Oreochromis corova, we have quite some diversity present, which actually would allow for the possibility that that species has been in the lake longer than we thought, or has invaded the lake with a higher diversity. Two possibilities here. So I did the same approach here. I pulled the diagnostic SNPs between those 4 species from an ND2 alignment, and then matched my fossil reads to that. And here the message is very clear. It's for this fossil, which is between 500 and 1,000 years old, we recover perfectly the Oreochromis hunteri sequence, so there's no doubt about that. And also for an older fossil where I have less coverage, but still where I have coverage, it's the Oreochromis hunteri sequence. So both species. Okay. 1 minute?
1 minute.
Okay. So yeah, so the method works, but no surprises. And Lake Victoria, I think I can do that very quickly because it doesn't. I was unsuccessful with ancient DNA extraction. It's a young lake, 15,000 years. It has a large radiation of cichlids, and we would like to know if there were any competitors, potential competitors present at the start. So it was dry about 15,000 years ago, so we see a paleosol in the course. And I do find fish fossils right after the refilling event, and mainly teeth, and there's a huge diversity of teeth, and they, although we don't get ancient DNA from them yet, we can already tell quite something by their morphology. So we do find the haplochromines, of course, which represent almost 500 species of the modern fauna. But we also, right after the refilling, we do find potential competitors, for example, these oreochromine teeth, which are also cichlids, but a different non-radiating lineage, and these cyprinid teeth. So we have evidence that right after the refilling, we have potential competitors, and it's not due to this ecological opportunity that the cichlid, that the haplochromines radiated, but apparently due to other factors. So the take-home message here really is that we can generate ancient DNA sequences also from tropical samples, which are several thousand years old, and I find that really cool. And I hope to answer other questions about allele frequency shifts over time, or the contribution of one lineage to another lineage instigating diversification, or how the pacing of speciation works better. And again, please note my collaborators on these projects and all the people that helped along the way, and also our funding agencies. Thanks.
Thank you very much for inviting me to present the results here. I would also like to thank all these collaborators mentioned here. This is part of a larger project in which several universities work together, also funding from the Arts and Humanities Research Council. Mary already hinted on this, that some of these techniques are very useful for understanding some archaeological sites. And I'd like to give an example of some of these sites, in particular some wetland sites where we can take sediment cores from lakes adjacent to these sites. Or actually, I should say, sites that are actually inside the lake, because that are the particular types of sites that I'll be showing. They're called crannochs. And so crannochs are small islands in lakes. They're predominantly found in Ireland and Scotland. And they are human built or human enhanced, let's say. So Often they're on promontories or in slightly higher points in lakes, so they're kind of natural features, but then they are enhanced, they're built up, often with a large timber framework. They put stones on top, clay and other kind of materials to make them larger, and actually large enough to also hold some structures. So there's quite a bit of archaeology done on some of these. And what is very common is they found this ring of like a palisade around the Kranos as well. And these are quite useful later on. I'll come back to that in a second for dating them. So these Kranos are quite common in some areas, and they were mainly built, let's say, in the period from the Bronze Age to the early medieval period, especially Iron Age and early medieval periods. And so what we did, so here's some examples of what they look actually today. So basically small islands in a lake with a lot of trees on them, but they're quite conspicuous, so they stand out on aerial photographs, and you can see them quite easily when you walk around lakes as well, so quite easy to spot. But there's some questions about them. So for example, it's very difficult because they're kind of beginning to disintegrate, overgrown. It's sometimes quite difficult to reach them. Some of it requires underwater archaeology and diving, which can be dangerous. And so not that much is known about them. For example, for how long were they used? Were they used repeatedly in different periods? Did people go back to these sites again and again? Also, can we find similarities between how these were constructed, between those in Scotland and those in Northern Ireland, for example? And then finally, that's something that I'll focus on mainly during this talk. So what was their function, actually? What were they used for? There's a lot of ideas about this. A lot of them to do with that they are part of structures that were built by let's say, the more well-off, the regional elites, because it takes quite a bit of coordination and effort to build these structures. And one of the questions is whether people actually you use them as kind of bolt holes in more, let's say, periods of unrest, when they also put, like say, cattle on there and other valuables. And so that was one of the things that triggered us to use some of the molecular techniques to also see if we can find evidence. But before I get to that, I'll just give you a quick overview of where you can find them. So there's a definite, let's say, Celtic distribution. You might say almost like non-Roman distribution or something like that. But they go back much longer, so some of them really are Neolithic as well. You can see quite a few of them are actually not dated, so only the red and orange dots are dated, and only the red ones have some kind of dendro or radiocarbon date on it, often only on one of those palisade poles. So whether they were built at the start or during one of these periods of occupation, maybe, this is still unclear. And so this is also one of the reasons to look at that. So I'll present you data from one site, Loch Yon in Northern Ireland, which is close to Enniskillen. And this is a site with a very small, well-contained catchment. So we don't expect to find a lot of things coming in from far away. We expect very local signal there. And you can see the lake, and there's actually two Kranos in the lake. One is very clear, and it stands out, it's right there in the north. And the other is here, which is now grown on to the mainland, but it used to be kind of probably separate from the land. And we took several cores, and I'll focus on the ones actually taken around here, where we did several analyses. This is our sediment sequence. Actually, it's just Lake Gitja, and then it's let's say, interbedded this bit with a lot of charcoal, animal bones, fragments, lots of wood, and so there's very clear stratigraphic layer of where people were adding things to this lake, probably just shoving things off the Kranoch into the lake, and so you get this kind of continuous build-up of material. In this particular Kranoch, originally there was only a date on this pile, which went back to, well, you can see the date there, about somewhere between 1000 and 1250 or something like that. But when we dated some of the material in this, we saw that actually this period stretches a bit of a longer period there. And this is very similar. This is from one of the other cores from the same lake, the other Kranoch. The start date is very similar to the start date here. So we thought, okay, we're probably on the right track. The chronology looks all right. So far, there's more dates coming, actually. One of the things we could see very clearly, if you look at some of the XRF data, we get an idea of this is a titanium record. So you can see there's areas in which there's a lot more minerogenic materials coming in. So probably this is to do with kind of cranach construction. People actually put clay and silt on top of that to extend it. And later on, you can see that it's probably breaking up. It's falling apart. It's eroding. So it's kind of spraying into the surrounding area. If we look at pollen, so I've kind of highlighted the main things to see, so it doesn't really matter so much that you can't see all the individual species, but I think the main thing to see here is that there's this period that coincides with, the start of this cranach building, where things like species like oak, corylus, ilex, holly are kind of declining quite steeply, and other species that are more indicative of like open meadows, grassland types, so plantago, lots of grasses, and also bracken actually, rumex, and you also begin to see this kind of cultivated. So this is pollen evidence, and then I said that already, and then we also have diatom data which show a very clear increase in nutrients kind of in the earlier phase of this cranach, and then there's a decrease again in nutrients, which looks at maybe the first half, let's say, of this phase is a bit more intensive use of these cranachs. One thing that really stood out in some other core, I'll just highlight it here, is that there's very high concentration of plantago, plantain, in here, and this is also what we see on the core that we took next to the other cranach. It's very similar kind of setup. So you see here the initial start of this cranach building, and you see this increase of various of these things. And the most interesting thing was that we found this also on some material that we analysed from the cranach itself. We took some samples and had a look, and this was absolutely packed with plantago. It was packed also with other things, but so charcoal was very abundant, obviously. And so likely this means that this plantago it probably hasn't grown on the cranach in that sense, but we think it's probably from dung that was brought, so animals were grazed around the cranach and then brought onto it, and the dung creates this high amount. The other thing that was very common on there was bracken, so sporangia of bracken, and so this is also something that must have been brought there, potentially as bedding for animals, or even as thatching for roofs, but this is still a bit debatable, I guess, which of these uses. So one of the things that we were really interested in is actually to see if we can get some idea of livestock there. So if we get a very local signal of which types of animals were there. So basically what we did, we used very similar approaches been talked about earlier. She also had a plant primer as well as a mammal primer. For doing that, I won't go into this in detail, but you can come and talk to me if you like afterwards. And so this is another core which was taken just right next to that first core that I showed you, and you can see the very same thing. You've got this initial bit of increase. There's stratigraphically quite a clear indication, and then this is what the plants from the DNA. The metabarcoding approach showed us, and it's very similar. So you go from initially a woodland with ilex and quercus and elm, at some point to this more open landscape meadow, which looks probably a bit like there in the top. Again, lots of plantagos common there, rumex, and you can see different grass taxa as well coming in, like Glyceria, holcus. And also very clear indication of various cultivars, so things like linseed, oats and rye are popping up as well. Right, so, okay, so I just highlighted plantago because it's in there, very common as well, and bracken. When we're looking at the wet species, there's not this very clear signal of eutrophication that we found using diatoms, but this is also because diatoms are such a good indicator for nutrients, and so we do seem to have some eutrophication going on, but it's not as clear a signal as for the diatoms. Now, finally, because this is what the whole what we really wanted to look at, and is to see if we could find evidence of various mammals, and we did. So from quite early on, there's some trace of cattle, but also others, so sheep. especially. So those are the two kind of dominant things that are appearing. Oops. But there's others as well. So we've got some evidence of wild boar or pigs, donkey or horse, goat, and also red deer. And so probably, actually, they might have actually also butchered things and brought it onto the Cranach. So this kind of begins to kind of piece together this image of what people were there. So we're fairly confident that this is actually a very local signal, which is something that Mary also indicated earlier on, where people just really literally chuck things off the Cranach in there, but we cannot at this point completely rule out that there's some catchment input as well. And so what really became clear is that there's this very strong coincidence of local presence of all these animals, livestock, with Cranach occupation. Although, and this is actually the red, so the red band indicates this is kind of cluster analysis based, so this indicates where, kind of statistically, is this different, let's say, set of samples. Although there is some indication that actually a little bit earlier, there's already a sign of kind of opening of the area. There is some hints of some of the animals as well, and actually maybe this initial little blip here in titanium, let's say, also is indicative that humans were already let's say messing about. So to conclude. I think there's several, in this kind of approach, you use several lines of evidence, obviously, both kind of, let's say, from a more geochemical approach and various paleoecological approaches, to kind of piece together this picture of what was going on at these sites, so to better understand the archaeology. So it seems people were actually bringing animals onto them, but we're still not quite sure whether this was just a seasonal thing. Plantago only flowers during a certain period, so you would actually need to have your animals grazing and then bringing them onto the Cranach to get these kind of large Plantago amounts. So this might say something about the timing in the year when people were doing that. It might actually mean that they did it all year round, rather than only in moments of dire need to find a safe space when there was local unrest. And then you see there's various cultivated crops as well that maybe they were processing there, or definitely in the catchment very locally. Soon we'll have better chronologies. They're kind of, we're waiting for them to come back, as well as comparing these with things like fecal sterols to get another indicator of human and animal input. So people at Newcastle, colleagues, they are currently analysing the fecal sterols, so the biomarkers, to see if we can find evidence of actual of the dung and whether it's human and whether it's animal or other animal, I should say, of course, and so other things as well. So I think this will then give you a complete overview of what was happening at such sites. And I think with that, I would like to thank again all the collaborating institutes and funders, and thank you for attention.
I'm glad that I have this opportunity to present this ongoing project that we have in Umeå, which is really north. So that's why it's easy for us to see Northern Light. I hope you are not too jealous. Right. So I changed the title a little bit. So we are now only focusing on reindeer and human hunters, but the preys. It's the relationship between preys and human hunters in the past. And we are trying to, because Laura previously, she said bacteria and plant DNA and animal DNA could be preserved in the sediments. But so far, there are not many studies looking into animal DNA in the sediments. So that's why we are thinking, okay, we are going to do some tests. So speaking of Sweden, I would like to show this figure, the Scandinavian ice sheet before Holocene. So there was a long time that Scandinavia was covered by ice, and till 12,000 years before present. And since then, the ice sheet became weak and it lost its volume, and then the size of the ice sheet just become really, really small. And then till 9.5 thousand years before present, there was almost just some dead ice in northern Scandinavia. So we are interested, so what's happening after ice retreat? It is believed that plants and animals, they came to Scandinavia after ice retreat. And this is the figure. This figure documents the two proposed routes of reindeer migration in Scandinavia. One is the route from south through Denmark to Sweden, and then to Norway, and then all the way to north. And the other direction is from Eastern Europe. So the migration of the other plant species and animals, they are more or less from these two directions. And however, our understanding of species migration hugely relies on the findings of fossils or pollens from archaeological sites with very much human activities. So that means there is a bias that if there were no much human activities, or if we don't find fossils, then we don't get much information about species and their distribution in the past. And this is why we want to do some tests in Sweden, because we know what time the environment become good enough for species to live. So in central northern Sweden, we have some research questions that we want to answer. And the first one is we want to know, can plant ancient DNA be preserved in lake sediments? Or in many cases, we know it works. And Sweden is like in really north. So ideally, environmental DNA should be quite well preserved. But however, in boreal regions in Sweden, we have mostly organic rich sediments. So that means we don't have that much minerals or clay particles to preserve DNA in the sediments. So we want to do some tests to see, okay, if we could find plant DNA first, because that's what we should find, and then And then second question is that we want to see if we could find ancient animal DNA, in this case, deers, because currently at the site, we could see reindeers and moose. So we want to know, okay, these two important species are now in Sweden. When did they come to Sweden? And we want to know if their DNA could be preserved in the sediments as well. And overall, the most important thing we want to understand is, okay, after ice retreat, who came to central northern Sweden first? Large terrestrial animals, in this case, moose or reindeer, or human hunters. And just in case some people don't know how moose look like, because I didn't know before I started this project. So this is the photo of reindeer and also a photo of moose. So they are very different. We have taken sediment cores from 4 different lakes in central and northern Sweden for different projects. However, we could only find animal DNA from this lake, Lake Christian, and this is a very specific site, so I will just focus on what we find from this lake. So Lake Christian is from central northern Sweden, and this is from the site that archaeologists found Stone Age human settlements and cooking pits, and also many burned moose bones. So that means they were hunters, and they came here to hunt moose, but not reindeer. So that kind of make us very curious, why only moose, or just because reindeer wasn't around, or there must be something. So, and this is just Ray. So this photo is just to show how we did excavations in 2010. And somewhere from this photo, we found cooking pit buried in the soil. And we also found the cooking stones that the early human used for cooking. And because we also found like very distinct charcoal layer. So we took the soil core from the soil profiler to also see if we could detect animal DNA from the soil as well. Even though, well, we tried and we did find animal DNA, the deer DNA, but we don't know if that's from mountain DNA or not, because it's known that there is always a leaching problem in soil samples. So I won't talk about that today. So overall, I have samples. I have different sample metrics. I have bone materials and also soil and sediments from the middle of Lake Clarke. And what we did is we had carbon-14 dating on the bone materials and the microfossils we found from the lake core to know the ages. And then we did DNA extractions from soil and sediments and many blanks by using mobile power soil kits, which is the most commonly used kits for DNA extraction from environmental samples. And then we had PCRs with primers amplifying plant and deer DNA. This is just a slide that we want to show. We had some controls to make sure we don't get much contaminations from sampling procedures or from analysis. So we had contamination tracers coating on the surface of the core, and then we brought the core back to the pre-sterilized sampling hood, and we had 1 or 2 person to remove the contaminated surface core, and then have another clean person to take subsamples across the stratigraphy. And then we brought the samples to a clean lab that we never did DNA analysis or amplification ever, and we just did DNA extraction there. And the results, so based on the carbon-14 dates on the burn bones, we found that the oldest bone material told us that the earliest Mesolithic human hunter arrived this area around 7.1 to 7.2 thousand years before present. And what we found from the sediments is, the first panel here is the stratigraphy of the sediment core, and you could see that the core contains the material from the pre-Holocene till, and also the material when the site was under marine environment, and also we have Holocene lake sediments. And the second panel here, just some basic chemistry of the sediments to show those sediments, the Holocene sediments, they are actually full of, the organic concentration is really high, which may influence the DNA preservation. And the most interesting thing is that the third panel here, we have, we show the frequencies of positive PCR with primers amplifying plant DNA, and also for deer. So surprisingly, we had plant DNA detected throughout the whole core, and that is something we didn't expect in the very beginning, because I didn't expect such well DNA preservation. And then with the deer, the primers targeting deer mitochondrial DNA, we found the oldest deer DNA was around 7.5 thousand years old, which is very close to the arrival time of early human. So we thought, well, so this ancient DNA as a proxy, it may tell us something more about what was the time, because maybe the prey or the other terrestrial species, they may arrive the site even earlier before human got there. And what we are doing now, well, because we tried to sequence the sample and we didn't get fragments of really good quality. So what we are trying to do now is to have more specific and short primers that will target DNA of moose but not deer, and then to see if we really could see, to check, to find the time where moose actually arrive central Sweden. And there's another thing I want to bring up, that the mobile power soil kit, well, it has been used quite a lot, but just a publication 2 years ago, they showed that actually this kit gave lowest DNA yield compared to the other extraction method. So that means we may only get, by using this kit or the other commercial kit, we may only get probably just 1/10 of the genetic information preserved in the lake sediments. So that means if we want to know a bigger picture about the ecosystem or species present or not present in the past, then we need a better method. So just to sum up, we found that the plant DNA could be well preserved in organic rich sediments for at least 9.5 thousand years in northern Sweden. And the ancient DNA from animals could be extracted from sediments in Lake Clarke because we could only find the DNA, animal DNA from this lake. So we cannot sure if that would apply for all of the lakes. And at this site, the estimated arrival time of prey is suggested by sedimentary DNA, which is 7.5 thousand years before present, and which coincides with arrival time of early human. And finally, I want to say, I think ancient mammal DNA in sediments with a more effective extraction method would help us quite a lot to understand the migration or the community in the past. Yeah, community of species in the past, even though fossils are rarely found. Thank you for your listening. And I would like to thank my collaborators and also the funding sources for supporting this project. Thank you.